My name is EmilyO'Hara. I'm a PhD student at JC University. I'm studying venomous jellyfish. So my focus is on one of the irukandji species here called Caricia banzai. I'm aiming to look at their venom to try and determine if their venom composition will change as temperature changes with environmental change. So as the waters around here are going to get warmer, is it going to affect how their venom changes? Because we know different things affect their venom, so different life stages affect their venom, things like that. So it's interesting to see if other ecological factors such as temperature will have an effect. We know the temperatures are going to get warmer in the seas with the projected climate changes, and it's important to know how their venom's going to change, because what their venom's made up of affects how it stings people and the severity of the sting. So being able to predict any changes will give you a really sort of preemptive approach to reacting to that. I finished my master's a couple of years ago. I took a couple of years off and then started my PhD about 6 months ago. So I'm in the very first year of my PhD. I'm just getting into the experimental phase now. When it's stinger season here, we go out on a night time and collect the irukandjis ourselves. So we do a lot of field work in that respect. We'll then use those animals to extract the venom from, and we process the samples like that, and that's how we get our venom. What the aim would be to do is we can use the wild-caught ones but also grow them up ourselves at the different temperatures I want to examine. So we then have them growing in those experimental temperatures, so we can then take the venom and analyse the venom like that. It's a bit tricky with this species. It's a box jellyfish, and box jellyfish in general seem to be very difficult to maintain in aquariums. What we have at the moment is jellyfish have two distinct life phases. They start off as a really small polyp that's attached to the substrate of the seafloor. They'll then metamorphose at some point into the swimming jellyfish that most people think of when they think of jellyfish. We have the polyps at the moment, and they're really easy to produce lots and lots of because they kind of clone themselves. They'll split off and you just get more that way. Trying to get them to metamorphose into the medusa is the tricky part, and that's kind of the first stage of my experiments before I can grow them up at the different temperatures. We are having some success at the moment. I'm midway through one of those experiments, yes, fingers crossed. And then the challenge is to work out how to keep them in the aquariums for a long enough period of time that we can get them acclimatized to the different temperatures and then take our samples, but it's looking promising at the moment. You can pretty much take the venom from any of the life stages. It just depends which life stage you want to examine. So what I'm hoping to do overall in my PhD is look at each of the different life stages through the different temperatures, because at the moment, especially the younger life stages, we don't really know what the venom does to people. You can't really record stings that way because they don't swim around. The ones we have at the moment, they take about 2 weeks to actually metamorphose, and then the tricky part is with the age. When we catch them in the wild, we catch the wild ones. We catch the older ones, so they're kind of juvenile and adult. We're sort of missing a life stage from the wild where they're very, very tiny. These animals only grow to about their bell size, are about this big anyway, which is why they're a problem here, because you can't see them, which is why people get stung. So it's a bit of we're not too sure how long it's going to take overall, but we do know they have to grow full size within a stinger season. So we're looking at periods of weeks to months here rather than anything longer than that. I've always been interested in marine biology. I did that at undergrad. From one of our undergrad lectures, I got really interested in the venom and toxins side of things. That sort of progressed into looking at sea anemone toxins through my master's, and then I kind of went on to, I'd like to look at some more medically important species, so the research could have a bit more relevance to the real world kind of thing. So it's gone from sea anemones to jellyfish. It's fantastic, especially the fact it's in my first year, so I can really get the extra things up and running off the ground from a really early start, which means, and like I said, we have those knowledge gaps in how long things are going to take to do and work out, so it's great that it's in my first year that I can use the money to put into my research and really get it going from the start.